Good morning and welcome, dear students. In respiratory physiology, the topic we move on to respiratory pigments. We are all known that organisms regularly utilize oxygen and release carbon dioxide in their attempt to liberate energy for the bodily activities. So respiration or gas exchange involves all such processes, including the exchange of gases and the biological oxidation of the absorbed food materials. So there are, from this physiologist standpoint, respiration involves two quite different processes: external respiration and the internal respiration. And the internal respiration mainly focus on the, or mainly known as cellular respiration. Internal respiration occurs within the cells and hence also known as cellular respiration. It involves metabolic reactions utilizing the oxygen and resulting in the liberation of carbon dioxide and energy. So this cellular respiration can be done with the help of certain respiratory pigments. So we discussed detailed about respiratory pigments. Respiratory pigments are conjugated proteins. Protein combined with metallic group, which gives a characteristic color on oxygenation. Conjugated proteins are protein. These are combined with any one of the metallic group, which gives a characteristic color on oxygenation. It imparts definite color to the body fluids. They consist of a protein coupled with a non-protein component, that is prosthetic group. The prosthetic group is a metal, usually iron or copper. There are mainly 4 substances are known to function as common respiratory pigments. These are hemoglobin, Hemocyanin, Hemoerythrin and Chloro Chrorine. These are the 4 important classes of respiratory pigments. And the oxygen binding site in various carrier protein. First figure is explained or illustrated protoporphyrin 9 in hemoglobin and myoglobin. This is the molecular structure of a respiratory pigment, hemoglobin and myoglobin. So in different respiratory pigment, oxygen binding sites are different. In protoporphyrin 9, it's a carrier protein attached in hemoglobin and myoglobin. And next is the figure second A illustrated iron core in Hemoerythrin. And the figure second B has illustrated copper core in Hemocyanin. So this figure clearly indicated that different types of metal or different types of prosthetic group, that is a metal, example iron or copper or protoporphyrin, are attached to the different regions of the respiratory pigment. So this helps to binding oxygen firmly for carrying the respiratory molecules, or it helps the cellular respiration. And the major functions of respiratory pigments are gas transport, mainly oxygen and carbon dioxide, and also gas storage, mainly the storage of oxygen, and maintaining the pH buffers, and also enhancing gradients for gas diffusion, and it also helping the non-respiratory transport, and it also helping enzymatic functions, etc. These are the general major functions of respiratory pigments in organisms. And the major respiratory pigment are hemoglobin. It's the most common type of blood pigment in vertebrate. It's present in the stroma of RBC in vertebrates. It is widely distributed. It is present in the stroma of vertebrate erythrocytes or RBC. Its oxygen carrying capacity is high compared to other pigments. It's an efficient vehicle for oxygen transport. It produced by liver cells, synthesized from acetic acid and glycine to form porphyrin, which combine with the iron to form heme molecule. So it is a metalloprotein. So hemoglobin is known as a metalloprotein. Metalloproteins are a form of conjugated protein. Each hemoglobin is made up of 4 tetrameric, that is 4 or tetrameric globular protein subunits. Each subunit or each monomer is composed of a polypeptide chain tightly associated with a non-protein prosthetic group called heme. So the heme is developed by the combination of acetic acid plus glycine, and the amino acid acetic acid glycine combine to form porphyrin. Then porphyrin is combined with a metallic group known as iron and formation of heme, that is 4 heme unit plus 1 globin chain gives hemoglobin. Each polypeptide chain has an alpha helical secondary structure in a globin fold arrangement. This is the formation of hemoglobin. Once again, acetic acid plus glycine combine to form porphyrin. Porphyrin plus iron gives to heme. 4 heme plus 1 globin combine to form a hemoglobin molecule. And the RBC, this is the combination of or this is the structure of hemoglobin. So in the RBCs, stroma of RBC, the hemoglobin molecules are combined. So 4 heme compounds are combined with and formation of a conjugated protein. This is the structure of hemoglobin. And this is the internal structure of heme molecule. And the specialities of hemoglobin are color of blood is due to heme. Heme is a combination of acetic acid plus glycine gives the production of porphyrin. Porphyrin plus iron gives heme, and 4 heme plus 1 globin is hemoglobin. So the color of blood is due to heme. In human, it's a tetrameric structure. Molecular weight in corpuscles is the 68,000 kilodalton. In Glycera and Notomatus species, 2 monomer with molecular weight 34,000 kilodalton. In vertebrate, hemoglobin differ in amino acid composition. with invertebrate arrangement and in oxygen binding characteristics. And this is the attachment or the surface of RBC. The stroma of RBC, hemoglobin is attached with RBC. So this contains to alpha chain, to beta chain, 4 iron molecules. So this is the helical shape of the polypeptide molecule. It's the hemoglobin, structure of a hemoglobin molecule. This folding of this polypeptide molecule contains a pocket which strongly binds the heme group. Here, each of the 4 heme portions of hemoglobin molecule contain 1 atom of iron. The iron is held in heterocyclic ring known as a porphyrin ring. The iron, which is the site of oxygen binding, the iron site is the oxygen binding site. Bond with the 4 nitrogen in the center of the ring, which all lie in one plane. The 5th bond position of the iron is bound strongly to the globular polypeptide. The 6th position can reversibly bind to 1 molecule of oxygen, completing the octahedral group of 6 ligands. This picture showing the detailed attachment of iron with the porphyrin and with the oxygen molecule. In the hemoglobin tetramer, the 4 polypeptide chains, 2 alpha chain and 2 beta chains are held together by non-covalent interactions such as hydrophobic interaction, ionic bond, hydrogen bond and polar bonds. The major covalent interactions are present in the hemoglobin molecules are with the alpha and beta chains are held together by the non-covalent interactions such as there are 4 types of bonds: hydrophobic interaction, ionic bond, hydrogen bond and polar bond. Overall, each hemoglobin molecule can combine with 4 oxygen molecule, that is 8 oxygen atom. And another condition known as effect of amino acid changing leads to a condition known as sickle cell anemia. In normal hemoglobin, its amino acid sequence is normal hemoglobin beta chain contain valine, histidine, leucine, threonine, proline, glutamic acid, and this is the amino acid sequences present in the beta chain, polypeptide beta chain. But in sickle cell anemic person or the person having sickle cell anemia, the hemoglobin beta chain has been altered. The normal hemoglobin beta chain contains 6th amino acid is the glutamic acid. So, in sickle cell anemic person, the 6th glutamic acid has been replaced by valine. So, this condition is known as sickle cell anemia. So, the RBC becomes sickle shaped and it doesn't carry the oxygen molecule. So, this is the figure illustrated the normal RBC and the sickle celled RBC. It's the normal hemoglobin beta chain amino acid sequence, and this is the in the sickle cell anemic person, the 6th glutamic acid replaced with valine leads to abnormal sickle shaped RBC. And the factors affecting affinity of oxygen with hemoglobin are specific variant of hemoglobin, cooperativity of binding in multimeric form, temperature, pH, carbon dioxide, organic phosphates, inorganic ions, etc. The affinity of hemoglobin under different conditions, and the conditions are by oxygen concentration high in the gas exchange surface. At that time, carbon dioxide concentration is low. At the time, affinity of hemoglobin for oxygen is high, result oxygen is attached. In the respiratory tissues, oxygen concentration is low, carbon dioxide concentration high. The affinity of hemoglobin for oxygen become reduced or low, the oxygen is released. This is the hemoglobin under different conditions. Next is the types of hemoglobin. There are different types of hemoglobin. In all these forms, the heme portion remains the same, where the globin subunit composition differ. So, globin composition has been differ. Hemoglobin variants are occurred. The human produce several types of hemoglobin. The HbA, HbA2, HbF and HbA1c. And the another one is the HbS. HbS is also known as sickle cell hemoglobin. So, HbA is the major hemoglobin in human. It is composed of 2 alpha and 2 beta chain. HbA2 is the, and also the polypeptide chain are 2 identical alpha and 2 identical beta chain. The alpha and beta chain have 141 and 146 amino acids residue respectively. More than 90% of the hemoglobin in adult human being is composed of hemoglobin A. And the hemoglobin A2. This variant form of hemoglobin contains 2 alpha chain and 2 delta chain instead of beta chain. Beta chain is delta chain. The 2 delta chain contain 146 amino acids, but 10 of them differ from normal beta chain. So, that is 10 normal beta chain HbA2 constitute about 2.5% the total hemoglobin content of the adult. And the next is the HbF. HbF is the normal fetal hemoglobin composed of 2 alpha and 2 gamma chains. The fetal hemoglobin has increased affinity for oxygen. The fetal hemoglobin is the main oxygen transport protein in the fetus during the last 7 months of development in the uterus. The fetal hemoglobin is nearly completely replaced by adult hemoglobin by approximately 12th week of postnatal life. Postnatal life delivery ക്ക് ശേഷം 12th week ആകുമ്പോഴത്തേക്ക് ഈ HbF റീപ്ലേസ് ചെയ്ത് പകരം മെച്ചർ ആയിട്ടുള്ള HbA അവിടെ വരും. ഇനി another one is the HbA1c. It has a glucose residues attached to beta globin chain. So, to alpha chain to beta chain, but one glucose molecule residue has been attached to the beta chain. So, such type is known as the HbA1c. And the another important one is the HbS. HbS is the, we discussed before, the HbS is a hemoglobin S or sickle cell hemoglobin. It is found in the sickle cell anemic patients. It consists of 2 alpha chain and also 2 modified beta chain, and the 1 beta chain has the glutamic acid in the 6th position has replaced by valine. So, this gives the RBC showing a sickle cell shape. So, in this condition, crescentic shape of RBC, the oxygen binding capacity of this person for sickle cell RBC is much very lower compared to normal RBC. This is the types of hemoglobin. And the next important respiratory pigment is the myoglobin. It's the iron and oxygen binding protein found in the muscle tissue of vertebrate in general and in almost all mammal. It is very related to hemoglobin. Found in the bloodstream is when it is released following muscle injury. It is represented as MB. Single chain globular. It is made up of single chain globular protein of 153 or 154 amino acid residues containing a heme prosthetic group in the center around which the remaining apoprotein fold. നേരത്തെ ഹീമോഗ്ലോബിനെ കണ്ട പോലെ തന്നെ ആൽഫ ചെയിൻ ബീറ്റ ചെയിൻ ഉണ്ട്, പക്ഷെ അതിൽ 154 അമിനോ ആസിഡ്സ് ആണുള്ളത്. ഇതേപോലെ ഹീം ഉണ്ട്. ഹീമും പോർഫയറിനൂടെ ചേർന്ന് അയണും പോർഫയറിനൂടെ ചേർന്ന് ഹീം ഉണ്ടായിരിക്കുന്നു. പിന്നെ പ്രോസ്തറ്റിക് ഗ്രൂപ്പ് സെൻട്രലിൽ വരുന്നുണ്ട്. പിന്നെ ഇതിനകത്ത് ബാക്കിയുള്ള അപ്പോപ്രോട്ടീനുകളെല്ലാം ഫോൾഡ് ചെയ്ത് കാണപ്പെടുന്നു. ഹാസ് 8 ആൽഫ ഹെലിസസ് ആൻഡ് എ ഹൈഡ്രോഫോബിക് കോർ. has a molecular weight of 60,700 Dalton. It's the first protein to have its three-dimensional structure revealed. അപ്പോൾ ഒരു 3-dimensional structure റിവീൽ ചെയ്തൊരു പ്രോട്ടീനാണ് മയോഗ്ലോബിൻ. ഇൻസ്റ്റൻറ്റ് ബൈൻഡിങ് ടെനാസിറ്റി ആണ് ഇതിനുള്ളത്. ബിക്കോസ് ടു ഓക്സിജൻ ഗിവൺ ഇറ്റ്സ് ഹൈപ്പർബോളിക് ഓക്സിജൻ ഡിസോസിയേഷൻ കർവ്. വളരെ പെട്ടെന്ന് തന്നെ ബൈൻഡ് ചെയ്യുന്നൊരു ഓക്സിജനുമായിട്ട് ബൈൻഡ് ചെയ്യാൻ എബിലിറ്റി ഉള്ളൊരു പ്രോട്ടീനാണ് മയോഗ്ലോബിൻ. High concentration of myoglobin in muscle cells allow organs to hold their breath longer. Longer breath നമ്മൾ എടുക്കുകയാണെങ്കിൽ മയോഗ്ലോബിനുമായിട്ട് ഹൈ കോൺസെൻട്രേഷൻ ഓഫ് മയോഗ്ലോബിൻ മസിൽസിൽ ഡെവലപ്പ് ചെയ്യുന്നു. അതേപോലെ ഡൈവിങ് മാമൽസായ വെയിൽസിലും സീഡ്സിലും മസിൽസിൽ ധാരാളം മയോഗ്ലോബിൻ കാണപ്പെടുന്നുണ്ട്. ദിസ് ഇസ് ദ സ്ട്രക്ചർ ഓഫ് മയോഗ്ലോബിൻ. ദ molecular structure. Next one is the hemocyanin, the main non-heme respiratory pigment. Its wide occurrence always present in the colloidal solution. It's the copper-bearing protein. It has a blue in color, found in the main phyla of invertebrate molluscs and arthropoda. Its molecular weight is 50,000 kilodalton. Arthropoda has 74,000 kilodalton. This is the molecular structure of hemocyanin. The molecular structure in a collar-closed octopus hemocyanin, and the collar is established. Figure showing the formation of octopus hemocyanin, and this is the gastropod hemocyanin. It's a cylinder shaped. And the arthropod hemocyanin is the molluscan hemocyanin. This is some difference in the functional subunits or in the active site. Then hemocyanin combined with the carbon monoxide leaves the carbon monoxide when in contact with air and regains its characteristic color. And next is the hemoerythrin. Hemoerythrin is the iron-bearing protein. So molecular weight is 10,000 kilodalton. It is found in the coelomocyte corpuscles and circulate with the coelomic fluid. When it is combined with oxygen, its color is purple, and detached with oxygen, it is colorless. It is found in only in few groups of animals. So molecular structure and the oxygen binding to hemoerythrin. Time of oxygen binding, it become purple color. At the dissociation, it become colorless. The structure of hemoerythrin. Next one is the Chloro Chrorine. It's the another oxygen binding heme protein. It is seen in annelids, mainly marine polychaete. Its oxygen affinity is weaker than that of most hemoglobin. It's a dichromatic compound. Appears green in dilute solution and red in concentrated solution. So it produces two different colors. So it's a dichromatic compound. Its structure is very similar to erythrochrorine. Erythrochrorine is likewise very similar to multiple subunits of myoglobin. Myoglobin പല സബ്യൂണിറ്റുമായിട്ട് സിമിലാരിറ്റി ഉള്ളതാണ് erythrochrorine. ആ എർത്രോ-ക്ലോറിനുമായിട്ട് വളരെ സാമ്യമുള്ള ഒരു പ്രോട്ടീനാണ് ക്ലോറോ ക്രോറിൻ. ഇറ്റ് കണ്ടെയ്ൻ 60 to 70 kilodalton myoglobin-like subunit arranged in a giant complex of over 100 subunits with interlinking protein as well as with a total weight exceeding 3600 kilodalton. Chloro Chrorine carries an abnormal heme group structure. It's found free floating in plasma and not contained within RBC. RBCയുടെ പുറത്ത് ഇത് കാണുന്നില്ല, ഇത് ഫ്രീ ആയിട്ട് പ്ലാസ്മയിൽ ഇങ്ങനെ ഒഴുകി നടക്കുന്നു, അതായിട്ടാണ് കാണപ്പെടുന്നത്. The molecular structure of Chloro Chrorine. And the expected question from these portions are: what is the role of respiratory pigments? How many types of respiratory pigments are there? What do all respiratory pigments have in common? What are the important respiratory pigments in arthropod? Explain the structure and functions of respiratory pigments.